the paper or the questions, I'll be writing on that. And then on the bottom screen, I've got our website, which is going to have all the notes. So I can use the notes to help explain everything. So yeah, try and answer the questions. If you do know anything, even if you don't know it, just let me know. I'll try and respond to you guys. And yeah, let's get started then. So first question, a single-celled organism has a large surface area to volume ratio. How does oxygen enter a single-celled organism? So this comes under transport in cells. So it'll be in diffusion. So what happens in diffusion, if you look at the notes, you can see it's a process of particles spreading from an area of high concentration to an area of low concentration. That's what happens whenever oxygen enters an organism or a cell. So the answer for the first one is diffusion. Okay, so the next one is a question about surface area to volume ratio. So we've got two cubes here. They both have different lengths. So this one has a length of 1. It doesn't say if it's 1 centimeter, 2 centimeters, or they're talking about meters, but it doesn't matter. So one's got a length of 1. The other one has a length of 2. You can't access the site, it's just loading. You might have a slow internet connection, so you just got to wait. Once it's all loaded, then the site will allow you to look at all the notes. Is this combined and triple? This is both. So the questions at the start are a bit easier, and as I go on, they start to get harder. It's relevant to both combined and triple. I've got a mix of everything here. Okay, so this one, yeah. So we've got two cubes. So on my notes, we've actually got this exact same example. So look at this one here. We've got one cube which is 1 meter by 1 meter, and another cube which is 2 meters by 2 meters. The question's asking, calculate the surface area to volume ratio of cube B. So cube B is this one here. So that's the 2 by 2 by 2 cube. So if we look at the notes here, this is how to work out surface area to volume ratio. First, you need to work out volume, and then you need to work out surface area. So what they've done here in this question is they've broken it down into 3 steps. So first you want to work out the surface area of one face of a cube. So one face is literally just a square. It's 2 by 2. So you just do 2 times 2, which is 4. So surface area of one face is 4. Then we got the surface area of the whole cube. So a cube has 6 faces. Thank you for sharing the live, Olivia. 6 faces. So what you need to do here, because the surface area of one face is 4, you need to do 4 times 6 to give you a total of 24 as the area. There's no need to put units here because they haven't given us any units. If you look at the question, they haven't given us any units, so you can just leave it as these numbers. Then to work out the volume. So to work out the volume of a cube, you want to do length times width times height. All of those are the same here. They're all 2 by 2 by 2. So just do 2 times 2 times 2, which gives you 8. So we got answer of 4 for the first one, 24 for the second one, and then 8 for the third one. So the surface area to volume ratio is 24 to 8. 24 to 8. Hope that makes sense. Let me know if there's anything you don't understand. Just let me know. So on to the next one. So we've got here an alveoli, which you can find in the lungs. Alveolus, singular alveolus. So what happens here is when you breathe in, oxygen comes in and it diffuses into these. So these are red blood cells. The red blood cell carries the oxygen, but at the same time, it gives out carbon dioxide. So carbon dioxide comes out and then the carbon dioxide leaves. So when you breathe in, it follows this green path. And when you breathe out, it follows the purple path. So the question says, oxygen passes from the alveolus into the blood. Name the part of the blood that carries the most oxygen. Anyone know the answer to that? Let me get that up on the notes while you guys try and think about the answer for that. Which part of the blood, there's 4 parts to the blood, which part carries oxygen? So that will be under organization, under the circulatory system. Yep, thank you. Red blood cells. So here, in our notes, we've got red blood cells are specialized to carry oxygen. You've got 4 things in the blood. We've got red blood cells, white blood cells, platelets, and plasma. Red blood cells are the ones that carry oxygen. So the answer is red blood cells. Well done, guys. Next question. Name the gas that passes out of the blood into the alveolus. So that's what I've labeled here. It's the purple line out of the blood. So that's this one here. Carbon dioxide. Well done, guys. Carbon dioxide. Next, alveoli provide large surface area for gas exchange. Give 2 other ways the lungs are adapted for efficient exchange. 2 ways that the lungs are adapted. Anyone know the answer to that one? It's to do with the shape of the alveolus. Actually, they've already mentioned the shape, haven't they? They've already mentioned that it's got large surface area. So you can't mention that. That's already been said. Anything else you can say? They've got very thin walls. So thin walls that are 1 cell thick. One cell thick. Yep, good. Not lots of mitochondria. That's in cells. I think you're thinking of muscle cells. They have lots of mitochondria. So that's one thing. Spongy, no, that's not going to make it more efficient. Good blood supply. Yep. So basically, if you look at a alveolus, you'll see that it has loads of blood vessels around it. That's because it's got a good blood supply. Moist lining. I don't think that'll get you a mark, but that is a good that's a good answer, but I don't think it gets you a mark for that one. That just helps the gas dissolve in the liquid around it. That doesn't improve diffusion, make it faster. Okay, good one. Next, question number 2. Water is lost from leaves of a plant through stomata. What is the process by which water is lost from the leaves of a plant? Well done. Transpiration. Yep, everyone's saying it. Answers transpiration. Good, that's another mark. So this question is all about transpiration. Usually like the first mark of a question, the first part of a question gives you an idea as to what the rest of the question is all about. So figure 1 here shows stomata on the lower surface of a leaf. So stomata are basically like they're holes that you have in the bottom of the leaf. These small holes which can change its size. They can open up and close. The name of the cell that basically opens and closes it, it controls the opening of the stomata, is guard cells. Well done, guys. You told me that without even seeing the options. Guard cell. Yeah, good. Next question. Okay, so we got calculation going on here. They've been kind, they've given us the equation here, but in biology, remember, you don't usually get equations. Biology and chemistry, you don't usually get equations. Only in physics they give you equations. So we've got a microscope question. So let's go on to our notes. Let's go into microscopes. Let's see if that can help us. So we got equation here. So that's the one that I was talking about. They've just rearranged it for us. Yeah. So all you need to do is sub the values in. But what you need to notice here is in the question, this is in millimeters. The question is asking in micrometers. So there's a conversion that we need to do. They've told us this, 1 millimeter is equal to 1,000. How do you convert? Here we have a conversion here. So to go from micrometers to millimeters, you want to divide by 1,000. And then the other way, you want to times by 1,000. Okay, we'll save this for the end. First, let's do the calculation. Size of image, it says, is 25 millimeters. So 25 millimeters divided by 800. You can convert it first. I see some of you guys are converting it now. Yeah, you do get equation sheet for physics, but in biology, you don't get an equation sheet. You don't get an equation sheet for biology, and for chemistry, all you get is a periodic table. For physics, that's the one where they give you a full equation sheet. So if you do 25 over 800, you should get 0.03125. This, remember, is in millimeters. We want to convert to micrometers. So to go from millimeters to micrometers, we want to times by 1,000. No, this is AQA. This is AQA. It came up a few years ago, I think. So 0.03125 times by 1,000. And yep, I can see most of you are getting it right. It's going to be 31.25 micrometers. Well done. Okay, let's look at the next question now. So a student measured the mass of water loss from some similar plants, water loss. So that's going to be all about transpiration. So what they've done here is they've got a plant. They've put a plastic bag around the plant. We'll talk about that in a second. They've put the whole thing on a balance, so they're weighing it. So what's going to happen? You've got the plant losing water. So water is like evaporating due to transpiration. So what you're going to expect to happen to the mass of the plant, you're going to expect this mass to decrease. You're going to expect it to decrease because it's losing water, it's going to lose mass. Good. So let's look at the method here. Seal the plastic bag around the pot of a potted plant. Yep, we can see that from the diagram. Place it on a balance at room temperature, 20 degrees. We'll come back to that in a second. Record the mass, and then you wait for 3 hours and then record the mass again. So what would you expect to happen? You would expect the whole thing to decrease. Calculate the mass of water lost, and then you repeat it at different temperatures. What was the independent variable in this investigation? What do you guys think? Independent variable, that's the one that you're changing. It's the one that you change. Dependent is the one you measure. And then you got control variable. These are all variables which you keep the same. Yeah, most of you are getting it right. So it's not mass. Mass is what you're measuring. So mass would be dependent variable. The thing you're changing, so look at the question. It says it's at 20 degrees first, then it goes to 25, then it goes to 30. So that's the one that you're changing. So the answer would be the temperature of the room. Suggest why the student sealed a plastic bag around the pot. Okay, so I told you we would come back to that. Why do you think there's a plastic bag around it? What does it say at the top, which you wrote? That says independent, you change. So this is what you change. You change. So nothing escapes. You need to give a tiny bit more information. Something, you're almost there. To avoid entry of water into the soil. Something else. You guys are close. It's basically to stop water from evaporating from the soil. Because there's water in the soil. We're trying to measure transpiration, isn't it? That's what we're trying to find out about in this question. So if we lose water in the soil, it's not going to be a fair test. We want to keep the moisture levels in the soil the same. Otherwise, it's not going to be an accurate experiment. So you can say, suggest why the students sealed a plastic bag around the pot so that water from the soil does not escape and decrease the mass. I'm not sure what year this is from. I think it's maybe 2018, 2019. Next one, the table shows the students' results. Okay, so let's try and look at these results. Mass at the start, so they're at 20 degrees, 25 degrees, 30 degrees. Every time there's a different mass. Then after they've weighed it, they've waited 3 hours and then they've weighed it again. This last column, that's a calculation. Mass of water lost. So what you're doing in this one is you're subtracting the two numbers. You're subtracting the initial number and the final number. Initial number, final number. First question, what is the resolution of the balance? Yes, most of you are getting wrong. You have a question yet? Just type it. If you've got a question, just type it. You don't need to say. How are we supposed to know how to calculate these questions when they never teach us? That is, try and go on my notes and see, because most of the information is on my notes. Or it should be in most textbooks as well, all that information. Resolution is 0.1. Yes, it is 0.1. Why is it 0.1? Resolution is basically the lowest reading that the apparatus can give. And you can usually tell it by looking at all the numbers. So you can see here, it's 508.6. 510.7. It's not 510. There's one decimal point after. So if it went all the way down to 0, it'll go 0, 0. That'll be the very lowest value. But then one above that, if the mass increases, it'll go to 0.1, because all these answers are to, all these readings are to one decimal place. So that means the resolution is 0.1, because 0.1 grams is the smallest reading of mass that you can give. And the question is, calculate the value of x in the table above. How do we calculate x? So I told you already, subtract this by this to get the last one. So to get the final answer, what you want to do is you want to add the mass lost to the final mass. Yep, most of you are getting it. Yep, George got it, Sean got it, Mosen's got it. Good, good, good. So what you want to do is 506.3 plus 4.9, which gives you an answer of 511.2 grams. Well done, those of you who got that. Give one conclusion that can be made from the results in the table above. One conclusion. So it's all about temperature. So you can say, as temperature increases, and then talk about this one here. So let's see what everyone's saying. As the temperature increases, the mass of the water lost in 3 hours in grams increase. Yep. So you can see here, the temperature, as it gets hotter, the mass of water lost is getting bigger, isn't it? It goes from, the temperature goes from 20 to 30. The mass lost goes from 2.1 to 4.9. So they're both increasing. So as temperature increases, the mass of water lost increases. Well done, guys. Nice. Give 2 factors that might affect the rate of water loss from the leaves. So I think I made a video on this a few days ago. The factors affecting transpiration. Thou. That's how you can remember it. So the first one is temperature. You can't mention that in this answer because it's we've already used temperature. This question is already all about temperature. So you can mention one of the other ones. So you can say, yep, the H is for humidity. The A is for airflow rate, which is basically just wind. And then the last one, yep, you guys are getting it, is light intensity. So one of those 4 should be acceptable. So yeah, let's put humidity and light intensity. HALT can be an acronym. Oh my God, that is so much better than thou. Why did I not think of that? That's actually a word. Thou is not even a word. That's better, guys. Use that. All right, next question. Pathogens are microorganisms that cause infectious disease. What type of pathogen causes malaria? No, not a virus. People always think it's a virus. Common mistake. Here, let me show you in the notes. I've got every single communicable disease you need to know about for AQA anyway. So go under communicable diseases. Let's go to types. We've got a table here of all the ones you need to know. This is the only one. Malaria is the only one that's a protist. Okay, viruses, there's 3 viruses you need to know, 1 fungus you need to know, 1 protist and 2 bacterial. Malaria is protist. So we got a question about malaria here. Give 2 methods used to prevent people catching malaria. That's also in the notes here. Prevention here, the last column. Nets, insecticides. We just need to give more information because it's a 4 marker. Saying the 2 won't get you enough marks, so we need to give a bit more information. So let's talk about insecticides first. So what does that do? They're going to kill the mosquitoes, aren't they? That's going to give you 2 marks. Insecticides, what are they? They're basically just it's the spray that you use, and it kills the insects basically. Nets, yep. Let's talk about nets then. So someone said nets prevent mosquitoes transferring. Yeah, basically they stop people getting bitten, right? Because if they get bitten, then that's how the protist gets transferred. Insecticides, you can say insect repellents as well. They're the ones that they cause mosquitoes to be less likely to bite you because they won't want to go near you if you've got insect repellent. There's both triple N combined. Injection, you can say vaccination. Don't say injection, say vaccination. Because what vaccination does would it'll make a person immune to it, so that'd be the second mark. But injection's not. It's not specific enough. Yeah. Thank you. Good to hear. All right. Next question. Describe 2 differences between a bacterial cell and a eukaryotic cell. So that's prokaryotic. This is eukaryotic. You guys tell me what you think the answers are. Let me get that up on our notes. Animals, the very first topic. Prokaryotic cells here. So one difference. No nucleus. Yes, good. And you can mention plasmids. Yep. You guys are getting it right. In fact, I watched your videos and aced my bio paper too. Amazing. I love hearing stuff like that. So, yeah, you guys are right. No nucleus. You have to say which one. So you can say bacterial cell. You can't just say no nucleus because you need to know which one has no nucleus. So bacterial cell has no nucleus. And you can also say bacterial cell cells have plasmids. Yeah, you can talk about size as well. The eukaryotic one is much bigger. Flagellum. In AQA, you don't learn about flagellum, but that is right. That is right. Okay, let's go on to the next one. Okay, so the questions are starting to get harder now. A scientist investigated the population growth of bacteria in a culture solution. At the start of the investigation, the culture solution contained nutrients, so that's basically food for the bacteria. The scientists determined the number of living bacteria cells in the solution every hour over 2 days. Every hour over 2 days. Figure 2 shows the apparatus. Why are there holes? Why are there holes here? What do you guys think? To prevent bacteria spreading? No. That would do the opposite actually. If it had holes in it, it probably would make that bacteria escape. No, they don't need moist conditions. There's a solution in there, so it's already moist inside. Yes, so that they can get oxygen for respiration. Good. Because they need to aerobically respire to survive, isn't it? So you can say to provide oxygen for respiration. That's all enough for 1 mark. Okay, this is a tricky one. So we got 4 stages here. So what they're doing is they're measuring how many bacteria there are every 2 hours, isn't it? So at the start, there's barely any. It starts to rise, then it levels off, and then it goes down. So for each stage, they want you to explain what is happening to the number of bacteria and why is that thing happening. Give one reason for what is happening. So the first thing, A, is basically just the very start where it's flat. So why is it flat? Because the bacteria haven't reproduced yet. Yeah. It's the very start. They haven't had a chance to reproduce. It'll take some time. Then stage B, it starts to increase. Yep, it's because they are rapidly reproducing. Why are they rapidly reproducing though? We need to give a bit more information because that's explaining the graph. So population rapidly increases. Why? Let's see what you guys are saying. Some of the bacteria, nope. Excess nutrients. Yeah, it's all to do with the nutrients. Yeah, they've got loads of food available. Remember, this is like it's a bottle, and the bottle is full of nutrients. So a lot of food. So when the bacteria are first introduced, they have all that food. So they're going to just eat it, and they're going to reproduce. So we can say because there is a large supply of nutrients. Is this triple? This is both triple and combined. I think it's relevant to both. I don't know if you guys have you guys downloaded the paper already? So like for our next lives, let me just show you. Just go to the website, and at the bottom here, yeah, these are all the lives that we're going to do. You can just download it. So this is today's one. We'll be holding one on Sunday, and then next week on TikTok we'll have one on Wednesday. You can download it. Much more useful if you have it in front of you while you come to these things. Yep, website is completely free. All the notes, all the questions, everything I'm showing you here is completely free. So yeah, go check it out after the live. Stage C. What's the name of the site? flashrevisionlab.co.uk. Here, everyone can screenshot this if you want to see. It's on our bio. So you can get notes, videos, and exam questions. Right, stage C. Thanks, Sean. So what's happening at stage C? So this is where it's staying completely constant, flat. So what's happened here is, yes, there's a lot of nutrients, but there comes a stage where the population is so large that the bacteria cells start to die. And the amount that are dying is the same as the new ones that are being created. So they're reproducing, they're dying, but the rate of both is the same. So the rate of cell division or reproduction is the same as the rate of death. Okay, so yep, so the last one, why they're dying? Because they die really quickly. Bacteria have really low life. They don't live very long. They can live for like a few seconds. We live for like 70-80 years. Bacteria, they reproduce very quickly and they die very quickly. So they will start dying eventually, but then the population size depends on how quickly they're reproducing as well. Can you not say that it's staying constant? No, because it's saying give a reason. So why is it constant? So saying it's constant doesn't actually get you the mark, because you need to give the reason of why it's constant. So you're meant to identify that it is constant and then explain why. Then for the last one, yes, the last one, there's not enough nutrients. That's why they start to go down, because now the nutrients are reducing because they're basically finishing off. Again, saying their population starts to decline is not enough for this, because that's describing the curve. It's asking for a reason. So you say the amount of nutrients decrease. Yeah. This is both triple and combined. It's relevant to both. Next. Okay, this is a bit of a calculation to do. It's quite a tricky one for biology, but I've shown you one similar already. So we've got two cubes. So let me go back to the notes where we had the two cubes. It's just awkward numbers. The calculation is the same as before, but the numbers are a bit awkward. So here's the examples of the calculations that we look at. So the question says, a bacteria cell can absorb all the nutrients it needs by diffusion through its outer surface. Explain why a multicellular organism cannot absorb all its nutrients by diffusion through its outer surface. You must include calculations. It's actually a very vague question. But hopefully, by the fact that they've given you two cubes, you can figure out that that's what they want to do. They want you to work out the surface area to volume ratio. Previous question, where did the nutrients come from? They were added. That was at the start of the question. They told us at the start of the questions that they added nutrients in the solution. That's where it came from. Back to this question. So surface area to volume ratio. So we got cube X, we got cube Y. Hope you guys got your calculators with you. This is AQA. All the questions here are AQA. So for cube X, we've got, let's look at this number properly, 2 times 10 to the 1, 2, 3, 4. So this is 2 times 10 to the minus 4 centimeters. Let's work with that. So for X, to work out the volume, you can do 2 times 10 to the minus 4 cubed. Can someone tell me what that is if you've got a calculator on you? And then surface area, you do 2 times 10 to the minus 4 squared to work out the area of one face, times by 6 because there's 6 faces. Okay, so we've got 8 times 10 to the minus 12. Thank you, guys. And for the bottom one, so that's 8 times 10 to the minus 12. And the bottom one is 2.4 times 10 to the minus 7. Okay, good. Okay, so these are really awkward numbers. So the surface area to volume ratio of X is 2.4 times 10 to the minus 7 to 8 times 10 to the minus 12. We can't leave it like this though. We need to simplify this ratio. We need to make one of the numbers 1. How do we make one of the numbers 1? It needs to be something to 1 or 1 to something. Divide both sides. Good. You want to divide both sides, not by surface area, whichever one's smaller. It's kind of like a maths. It's a maths topic. Ratios. You divide it by the smallest of the two. So out of these two, the smallest one is this one because it's to the power of minus 12. So the right hand side you need to divide by 8 times 10 to the minus 12. And left side you need to divide by 8 times 10 to the minus 12. Okay, so let me repeat that. If you've got a ratio, you want to simplify a ratio, you want to divide by the smallest number out of the two numbers. It's not always surface area, it's not always volume, it depends whichever one's smaller. What topic is this? This comes under diffusion. So, what do you get? So for this side, it's going to be 8 times 10 to the minus 12 divided by 8 times 10 to the minus 12. That's just 1. That's just 1. Because you're dividing it by itself. Yeah, biology. On this side, can someone do that? So 2.4 times 10 to the minus 7 divided by 8 times 10 to the minus 12. What is this number divided by that number? Anyone got the value there? You got 3,000. You sure? Look at the numbers again. I think it should be 30,000. 30,000. Just double check that. Check the number of zeros. There should be 4 zeros, not 3. Yeah, 30,000. So the ratio of the one on the left is 30,000 to 1. Okay. Now we need to do the same for Y. This is a long question. 5 marks though. Same for Y. So volume, let's work out the volume and surface area. But we've got easier numbers to work with this time. It's just 11. So we do 11 cubed here. Then we got 11 squared times by 6 faces. You would skip this. Go to it. Go to it at the end. 11 cubed would be, anyone got that? So 11 cubed is, no, this is AQA. AQA. Thank you. 1331. And then 11 squared times 6. So like I said, guys, the questions get harder in these papers that I make. 726. Thank you, guys. So surface area to volume ratio is 726 to 1331. And what did I say? How do you simplify this again? What did I say? How do we simplify this ratio? Divide by the smallest. Good. So this time, which one's the smallest? The smallest value is the 726. So you want to divide both sides by 726. 726. So when you do 726 divided by 726, that's going to be 1. And on the other one, actually, no, we need to make a comparison, isn't it? Okay, fine. We need to make a comparison because this is something to 1. We need to get this something to 1. Sorry, 1 second. So if you just wanted to simplify it, yes, you can just divide it by the smallest. But because we're making a comparison, we need to do the same as what we did here. We divided by the volume here. We need to divide by the volume here as well, just so we can make a comparison between the two. Because this question is asking us to compare the one on the left to the one on the right. So divide both sides by 1331. Never seen a question on this. This is either on the 2018 or 2019 paper. I guarantee you'll see it. AQA. So 1331 divided by 1331 is 1. And then you do 726 divided by 1331. Yes, you do get a decimal. That is fine. 0.54. That is fine to have a decimal. Okay, so the key thing here is you can see here the surface area to volume ratio of X of this smaller cube is much greater. Look at that, that's 30,000. This is less than 1. This is 0.54. So we need to explain that for a few extra marks. You can say that because this one has a lower, so we can say, so the Y represents multicellular organism, X represents bacteria. So you can say for the multicellular organism, have, what is this spelling? Multicellular organisms have a smaller surface area to volume ratio, so diffusion is faster. That's the last point to make, because that's what the question's asking. Explain why multicellular organisms cannot absorb all they need by diffusion. Basically, diffusion is slower. Diffusion is slower. So there's less diffusion. So because there's less diffusion, it means they can't absorb all the nutrients they need. Right. Let's answer some questions here. I know that was a complicated question. I thought we divide by the smaller number. Okay, yeah. So, like I said, guys, yes, you do. If the question just asked to work out the surface area to volume ratio, you divide by the smallest number to get 1 to something. Yeah. But because this was meant to be a comparison, we wanted to compare this number, this ratio to this ratio. We wanted it to be a number to 1. We wanted the right-hand number to be 1. So what we did is we divided by the right-hand number every time so that we got 1. That's why we did that. Would you suggest The notes for Cambridge IGCSE, yeah, yes. Although you probably wouldn't, yeah, you'll definitely need a textbook for IGCSE because they have loads of different topics as well. But it covers, I'd say maybe like 60% of the IGCSE syllabus. This is biology GCSE. Biology GCSE. Okay, I think that was the hardest question on this. Let me see what else we got. Okay, fine. Next question. Okay, hopefully this is a bit easier. We've got another magnification question here. So we're going to use our magnification equation. We've got the magnification here, and we've got the real length. We got our equation. Can someone tell me what the equation for magnification is? Let me get it up in the notes. So it comes under cell structure, microscopes. So in the first question, they gave us this equation. This question, they haven't. So magnification is equal to size of image over actual size or real size. Yeah, that's fine. We want to find the real length. So we want to work out this. We want to work out A. So we want to rearrange this equation. So if you rearrange that equation, let me give you the triangle. It'll be the I at the top, M and A at the bottom. So if we wanted to work out A, actual size, or the real length, is equal to image size over magnification. Yeah, well done guys. So let's put those numbers in. Remember, this is a 4 marker. That's because there is a conversion here. We've got our number in millimeters. We need to convert it into micrometers. So remember, the conversion is this. Millimeters to micrometers, you need to times by 1,000. So image size is 43. 43 divided by 50. That's what you got to do. That gives you, you can convert it at the start as well. That's fine. You can do 43 times 1,000 and then divide by 50. Or you can do 43 divided by 50 and then times by 1,000. Either way, you'll get the same mark. So you get 0.86 millimeters. And then you do 0.86 times by 1,000 to get you your final answer, which is, yep, well done, 860 micrometers. Wait, what are we converting? We're converting. So you see here, the question is in millimeters and they're asking us in micrometers. So you need to convert millimeters to micrometers, and that's what we've got here at the bottom. To go from millimeters to micrometers, you want to times by thousands. Okay. Next question. Okay, so this is another question about microscope. So a microscope has a ×5 eyepiece lens. How can you use this microscope to observe a prepared slide of root hair cells at magnification? Right, magnification of 50. Okay, we'll remember that as well. This is a 4 marker. So in the notes, let me show you. I've got all the parts of a microscope here. And then I've got all the steps of how to use a microscope. That's what this question is asking, basically how to use a microscope. So let's copy. Yep, that's a practical. You got all the required practicals on the notes. You can just search required practicals at the top and it'll come up. So first step, you can say secure the slide on the stage. Then we've got lowest power. That's another point. lowest power objective lens. The objective lens, let me show you the difference. There's two lenses you've got here. The top, you've got the eyepiece lens. You can't change the eyepiece lens. That always stays the same. Objective lens, you can see here, there's three. This one you can change. There's three different ones. It gets higher and higher. So you always want to start with the lowest power objective lens, whatever that is. Then what you do is you turn the coarse adjustment knob to we'll say lift the stage as close as possible. Oh, okay, let me answer that question. The staining, yes, you do need to stain. We do have that in the notes. That's over here. That's preparing the slide. That's where you stain it. This question's not asking about preparing it for onion skin. This one's asking, how do you use the microscope to prepare it? How to use the microscope? Yeah, it's not asking you to prepare the slide. So it's just asking the second part. So once you've got the slide prepared. So we're assuming in this question that we've already stained the onion skin and we've got it on the stage. Next, we got look through the eyepiece and focus with the coarse adjustment knob. And then the last point, okay, so this is the hardest thing to get because it's asking magnification of times 50. The eyepiece is times 5. Okay, remember I said this you can't change. You can't change the eyepiece lens magnification. So what do you think the other lens, so the objective lens, should be to get a times 50 magnification? So the eyepiece lens is times 5. You want times 50. So what does the other lens need to be? Yep, thank you. Times 10. Because 5 times 10 is equal to 50. So you need to basically, on this last step, because we want greater detail, we want to switch to a higher powered objective lens. You need to say what power that needs to be. So switch to the times 10 power objective lens. And then refocus. Yes, it is difficult. Like I said, guys, the first few questions are easier. It gets harder and harder as we go on. But yeah, I've got another live on Sunday where we're going through chemistry and physics, if you guys want to join. And then we've got another TikTok, a free one, next Wednesday. But for those who do want to join the Sunday one, I've got a code here, FRL20. Use that promo code and you'll get 20% off for this Sunday's one. But the TikTok one next Wednesday, that's a chemistry one, that'll be free. So take a screenshot of that. It's only for the first 5 people that join though. So if you want to join it, then I think there's like 12 spaces for this Sunday. So if you want to join that, then use that code. Okay, next question. Root hair cells absorb water and minerals from the soil. A scientist investigated the rate of nitrate ion uptake by the 2 seedlings. And what they've done is they determine the mass of the nitrate ions absorbed by each seedling every 30 minutes for 4 hours. So what's happening here? We've got a table. This is ions being absorbed through the soil. Anyone know what process ions get absorbed through the soil by? You want to see the previous question? Okay, quickly here. Take a screenshot of that while I go through the notes. Yes, active transport. This question is all about active transport. Well done. So let's get that up. That comes under transport for cells. Active transport, here we go. We've got the notes here. So minerals, root hair cells use active transport to take in minerals. So what's happening here? Describe the changes in the rate of absorption for seedlings with no oxygen added. So they're talking about this column here. What is happening here? It's asking to just describe. It's not asking to explain. Just describe. So look at those numbers. What's happening? How many marks? It's 3 marks. So what can you say? You can say it increases initially. Yep. The rate is basically the fastest at the start. So it's not asking what happens to the mass. It's asking what's happening to the rate. So the speed at which the mass changes. So at the start, can you see it's going from 0 to 60. Then the difference decreases. It goes from 60 to 95. Then it decreases again from 95 to 105. And then here it's barely changing at the bottom. So what's happening is the rate is decreasing. So in the first 0.0 to 0.5 hours, so 0 to 0.5 hours, the rate decreases. That's 1 mark. Then, so remember to use numbers as well, because sometimes you get marks for stating the numbers. Then it decreases. We've already said that. So it decreases. Then it's constant. Yep. Good. It levels off basically. And then we've got to give a number for that as well. So the rate becomes constant. At what time does it become constant? Okay, people are getting confused. It's increasing. No, so what's increasing is the mass. Yes, the mass is increasing. The question is not asking about mass. It's asking about the rate. So the rate is the difference in mass. So yes, the whole mass is increasing overall, but the rate is the difference. So you see here, the difference between this and this is 60. Then the difference between this and this is 35. Then the difference between this and this is 20. So it's basically decreasing. The rate is the difference in mass. So this question is kind of here to trick you. So that's why we say the rate decreases, but the rate becomes constant at around here. Can you guys see the rate is barely changing? It's staying around 130, isn't it? So you could say either after 3 hours or 3.5 hours. Around 3.5 hours. Yeah. Good. I think this is, yep, we're on to the last question. Okay, so now we've described it. Explain what the results of the table show about nitrate ions, how nitrate ions are absorbed. Okay, so we've already stated that it's active transport, isn't it? That is probably 1 mark. Look at the notes below. Root hair cells use active transport to take in minerals. Don't confuse this with osmosis, because root hair cells use osmosis to take in water. Look at that. And then they use active transport to take in minerals. Let me write this here. So roots, osmosis, that's all about water. to take in water from the soil, but they use active transport to take in minerals. People usually get confused between the two. So the question is all about active transport. Does anyone know how is oxygen related to active transport? So one thing, well, why don't you guys answer that while I write this down? What about transpiration? No, this question has nothing to do with transpiration because it's nothing to do with water being lost. This is all about ions being taken up through the soil. So you don't need to mention transpiration. Yeah, so it's all to do with oxygen. So when it comes to active transport, let me show you in the notes. Active transport requires respiration, and respiration occurs due to oxygen, aerobic respiration. So in our table here, can you guys see with oxygen, so that's this one here, can you guys see the values are much bigger compared to no oxygen? So the left column is much bigger than the right column. That's because there's oxygen. And when there's oxygen, that means there's more aerobic respiration. And if there's more aerobic respiration, that means there's more active transport. So to get 3 marks here, is you say that more ions are absorbed in the presence of oxygen. Reason for that is because they are absorbed by active transport and that requires energy from respiration. Yep. Good. All right, on to the last question. So before we go on to the last one, guys, remember, if you want to join the live on Sunday, here's a code you guys can use to get 20% off. But I'll be live on TikTok next week as well. But if you just want some extra ones, on this Sunday, I'll be going through chemistry and physics. Nitrate ions are essential for plants to grow. Describe how nitrate ions are used in a plant to help the plant grow. Anyone know this answer? This is one of those very small points that you're expected to know. I do have them in the notes. Let's see if anyone knows this. What time on Sunday? It'll be from 6 to 8. 6 to 8. So it's 2 hours. It's a longer live. We're covering 2 sciences. I think there's 12 spaces left. And yeah, there's a promo code. I think there's five promo codes you can use. So the first 5 people will get it. So it helps it grow. How does it help it grow? I don't think anyone's got it right. Oh, yeah. I think Tupac's got it right. It's under photosynthesis. Here we go. How plants use glucose from photosynthesis. We've got it here. One small point. Glucose is combined with nitrate ions from the soil to make amino acids, which are then assembled into proteins for growth and repair. That's how you get your marks here. So 1 for saying nitrate ions combine with glucose. second mark is to form amino acids, which are used to make proteins for growth. What book am I using? This is our website, guys, flashrevisionlab.co.uk. It's not a book. It's completely free to use. Everyone can use this. I've basically gone through every topic in GCSE science, and I've made notes on them with diagrams. So you're free to use it. It's got notes, and then look, you can click on questions, and you can check these questions, all these different questions that have come up in the last few years. And you can look at the mark scheme. You can try them out yourselves. So every time you do a question, you can check them. Right now it's got combined topics, which is included in triple. But in triple, you've got like extra topics to do. So everything in it is relevant for triple, but for triple, you'd need to know a bit more. It's completely free. Yep, completely free. So yeah, you guys can use this. You can even look at the spec points to see exactly what you need to know from the exam board. And like I said, for all our lives, if you want to work, if you want to join any one of our lives, every Wednesdays and Sundays. So the Wednesdays ones are going to be on TikTok. They're completely free. So that's today's one. We got one on Sunday for chemistry and physics. You can download the paper before you come. That's probably useful if you look at the paper before you come, but it doesn't matter. And then next week we'll be doing chemistry on TikTok on Wednesday. So yeah, thank you. Good to hear the code. You're asking for the code. Here's the code, guys. Remember, it's only for the first 5 people. If it says it's run out, then just DM me. I'll give you another code.